Hello again, invertebrate enthusiasts. We're here with the last kind of, I promise, 5 to 10. Somewhere between 5 to 10, no more. I have a timer going. Summing up of some of the exciting things about velvet worms, the onychophorans. I remember we talked about them as having 13 to 14 pairs of lopopod legs, each tipped with this elementally constituent claw. And that has a lot to do with this story and how hallucinogenic drugs and paleontology in the 1970s and 80s combined with discoveries made in Canada in the in Yoho National Park, in the Burgess Shale, in the last century, all mix up together in our understanding, our contemporary understanding of onychophoran or velvet worm evolution and how that fits within our understanding of the panarthropods. Here we go. Just used up like a minute just talking about that. This is where we left off, talking about these amazing Chilean physicists and biologists who talked about hydrodynamic instability and the garden hose variety necessity involving the slime jets. Some amazing things and some crazy things have been published recently about onychophorans. And I talked about how, in the lecture, I talked about how tardigrades had appeared in a Proceedings of the National Academy of Science publication that thought that 18% of their genes had been derived from horizontal gene transfer from bacteria, and that's how they became these crazy, amazing things. And then within 9 days, there was another publication that said, no, you've got contamination. I have an example here for the onychophorans as well. This crazy publication on the top, caterpillars evolved from onychophorans by hybridogenesis, essentially saying an ancestral lepidopteran bred with, successfully, onychophoran, and that's where the morphology of the caterpillar comes from. Within several weeks and in the same journal, the quite right correction, caterpillars did not evolve from onychophorans by hybridogenesis. So it's kind of another example of rapid publication in a big high-profile journal like PNAS, and then a correction, a natural scientific correction when there's a disagreement and the evidence does not support the claims in the initial publication. So, to keep talking about the onychophorans, we're going to talk about the Burgess Shale. And I've mentioned the Burgess Shale a couple of times before, and probably you've had the opportunity to explore it. If not, what I'd like you to think about the fact is, if you consider your arm, and out here at the tip, you got your fingernail, and the length of however long your fingernail is, particularly if it's like short like mine, that represents in the length of history of life, multicellular life on the planet, that represents where humans have lived. But here we're talking about kind of the base of your hand, the Cambrian and the Precambrian period, and the Burgess Shale. So in terms of, if this is the age of our planet, we have the explosion of multicellular life here, and then all of human evolution and culture existing just in the fingernail and the detritus captured at the tip of your fingernail. So we're talking about an explosion of life that happened where almost all of the literally all of the phyla that we know in the Dunn et al. Phylogeny, they all appeared. We went from having mostly jelliforms, jelly-like life pre the Precambrian explosion, and then this explosion of life where we see all of the genera and species representing all of the remaining phyla, and some that we don't see anymore, including trilobites, which is a panarthropod group that no longer exists and at one point dominated life on the planet. So what caused this amazing explosion? There's different hypotheses, but one of them has to do with, there's a poster child for this explosion of life in the Precambrian, at the end of the Precambrian, of the Burgess Shale, and that's hallucigenia, which is this crazy-like animal that resisted classification for years and years and years, because literally we didn't know which way to place it, up or down. And this is a drawing that appeared in a book, an amazing book that I'll give you a link to later on, by Stephen Jay Gould in the 1990s. I loved that book, if you're interested at all in the evolution of life and diversity on the planet, and just good reading about biological things, Gould, who's passed on and has been dead for, it might be close to a decade or more now, But this book is just a wonderful capturing of enthusiasm of characters and of biological understanding of diversity on the planet. Now, when Charles Wallacott discovered the Burgess Shale in Yoho National Park in Canada, many of those fossils came back to the Royal Ontario Museum in Ontario, here in Toronto, and they resisted classification for decades and decades, and hallucigenia was one of them. Now, the Burgess Shale was a particularly good, this is a particularly well-preserved part of life because it seems like it existed at this kind of a cliff face where there was a rapid slide and then very rapid compaction and lithification of life that happened. And so soft parts that would not have been preserved in other areas were preserved in the Burgess Shale. And so we have this incredible window into life in the Middle Cambrian. And like I said, it exists in the Walcott Quarry in Yoho National Park. And you're looking at two of these principal paleontologists in the 1980s. Brits, both of them, Simon Conway Morris on the left and Derek Briggs on the right. Conway Morris is at Cambridge now, and I think Briggs is at Bristol University in the UK. And they, on the left, Conway Morris was the one who described hallucigenia, and he chose, Gould called it, a most unusual and truly lovely designation for his strangest discovery. He called it hallucigenia, to honor the bizarre and dreamlike appearance of the animal, and Gould suggests, based on some of the other life habits of Conway Morris, that as a memorial to an unlamented age, well, lamented by Conway Morris, to an age of social experimentation. The drug-like nature, inspired by a drug-like vision, is what he was suggesting. We're going to take a look at some of the link that'll be on the bottom, but it's some of Attenborough exploring some of these fossils.
This is one of the oddest of the fossils from the Burgess Shales. It seems to have 5 legs along the bottom and curious kind of lobes along the top, which presumably were some devices which help it to feed. But what kind of animal is that with 5 walking legs and feeding lobes along the top of its back? It was such an extraordinary thought that the scientist who described it thought it was a kind of hallucination, and he called it hallucigenia. But since then, more specimens have shown that in fact this is probably the wrong way up, and that it was really like that.
And that's kind of an amazing recalibration, that the spike-like legs that we saw, and you saw displayed in that drawing, are in fact facing the wrong way. That these are not spike-like legs, these are spike-like dermal projections. And in fact, those lopopod, what were presumed to be lopopod feeding tubes, in fact are legs, lopopod legs, like we see in the extant onychophorans. So, it might have looked something like this. Now, that suggests an evolutionary connection between one of these oldest forms of multicellular life, the hallucigenia, to an extant lineage distributed across the world today in the cold, wet, moist tropical forests, the onychophorans, or the velvet worms. One was marine, all of life was marine at the time of the Burgess Shale. One is obligatorily terrestrial. Now, today. And what we see in this recent Nature publication is a further examination, not just of the lopopod-like legs, but of the claws that tip each one of those lophopods. Now, you remember we talked about the onychophorans being characterized by the stacked construction of constituent elements, or the traffic cones, one on top of each other. So what you're looking at here on the left-hand side are slices through fossils of hallucigenia from the Burgess Shale that are kept in the Royal Ontario Museum. And what we see for B, C, and D, these are hallucigenia, and we see the exact same kind of stacked constituent elements that we see in E for the onychophoran, for the extant onychophoran. And this combination of microscopy, of fossils, and of extant living organisms suggests that this stacked element of sclerites in the claws is a unifying synapomorphy that groups the extant onychophorans from around the world, the ones I lust after and want to find and describe and be awed by in the forests of Costa Rica, with this marine hallucinogenic organism that baffled scientists for literally up to close to 200 years before our proper understanding was achieved. Now, one of the neat things that's happened, this happened relatively like 7-8 years ago, this understanding. More recently, 6 years ago, there was the same kind of slicing and dicing was done of the teeth. And what they found was that there are different onychophoran hallucigenia teeth here that resemble cross-sections, dental cross-sections of teeth across all of the ecdysozoans. So this is hallucigenia not just being now an outlier that makes us question how life evolved on Earth and we name after a hallucinogen-inspired vision, but now cross-sections of claws connect them to an extant living organism, and cross-sections of teeth actually provide a unifying, maybe the only unifying morphological character that links all of the ecdysozoans. So not just the onychophorans, but all of these ecdysozoans that formerly have been linked quite strongly by all DNA evidence and by molting. Molting appears here and there. It's not all the way throughout, but this seems to be molting, and this kind of cone-like structure of the teeth takes hallucigenia and makes it a critical element that links all things within the ecdysozoa. Amazing. And so these fossils show us that these abundant marine relatives of the onychophorans lived 550 million years ago, might have been one of the first things to migrate to land. And if you're interested at all in this era, I would really strongly recommend this book. And I found when I was trying to inspire you or give you some, like an Amazon link, there is actually a PDF copy online where you can go and read it and put it on your phone and read it at your leisure. And then finally, I'll also encourage you to go out with this as your example. Go out and visit your Royal Ontario Museum. Go out and visit your national parks and give them both money for your visits, because these are institutions that deserve our support. These are a slight video or a slideshow of visits of the Royal Ontario Museum after Conway Morris and Briggs went that go and sample, and they go there regularly to sample the Burgess Shale and to discover more about the evolution of multicellular life. And I say it to you explicitly because I've seen people act on it. And Rebecca Smith was a student of this course in 2016. She's now doing a PhD at the University of Victoria, and she visited the Burgess Shale in the summer after she took this course, and she is showing the delight at finding some of those fossils as she was let into this global amazing position. So, 12 minutes. All right. I love this group. All right, we'll talk to you on Wednesday.